I'm Gerhard Pietersen, and my colleague Kerstin Krüger will be telling you about how to collect samples for Aster Yellows tests. This is part of a project, an international project, where we're looking at 3 different diseases, which includes Aster Yellows, with the project's called Tropisafe, and it received funding from the European Union Horizon 2020 call. In this presentation, we're going to be looking at collecting infected planting material for tests for Aster Yellows. We'll be looking first at symptoms, the time of year to collect, tissue to collect, records that you should take, the submission of the material for tests, and then mapping. And then slightly more on a research note, if we were looking for the vectors, how to do sweeping and to use a putter, vacuum collection with a DVAC, and sticky traps. We'll also look at sampling for potentially alternate hosts of Aster Yellows, as well as collecting of potentially new vectors of this same disease. To start off with, the symptoms are most prominent in early summer, late spring, where you get this yellowing on white cultivars. The leaves become quite brittle and leathery, and this is it can be confused with a number of other symptoms, and therefore it's important that we collect this material for actual lab tests. So once again, because the symptoms are best at this time and the pathogen is in fairly high titer, late spring and early summer is the best time to collect this material, and we recommend that only symptomatic leaf and PTL material be collected. This must be placed in a plastic bag. Here we have a plastic bag with the accession number for that specific plant, and this has to be kept cool, basically between 4 and about 10 degrees until it gets tested for the Aster Yellows phytoplasma. You should be recording the symptoms that you observe, the GPS position of the infected vine. It's always a good idea to also make a note of the row and the plant position of the infected plant in that vine. Good to have the date, the cultivar, the location, who the grower is, the sample number, and then who collected it. All this information is very valuable for later work on this pathogen. You can also use the row number and plant position as a coordinate system, where in Excel you just punch in these 2 coordinates as X and Y coordinates in a scatter plot, and this allows you to determine the spatial position of infected material. This is very valuable epidemiological information to obtain. To collect the vector, the best method is to do sweeping with an insect net, and then within that net to collect the sample using a putter. A putter is basically a vacuum, and on the one hand you draw up the insect and you create the vacuum on the other end. So basically just by sucking on this end, you will draw up the insect into the bottle. It's a good idea to have a little filter there so that you're not sucking up insects into your mouth. In this way, in the sweeping net, you can capture this little guy, Mgenia, who's the main vector for this pathogen. To monitor for the vector, sticky traps are hung out in the vineyard for different periods, sometimes up to 2 weeks, sometimes a month, sometimes only days, depending on the purpose of the monitoring or the accuracy of the monitoring. The little leaf hoppers get stuck on this. They're attracted to the yellow color and they get stuck onto the trap. The little sticky trap is then covered in a plastic bag where the insects can then be counted, identified and counted. Here's an example of how we do a collection. First of all, taking a photograph, you'll see we have a pole there to allow us to determine scale of the plant. Then in this case, we're collecting any plant material because it's asymptomatic. We don't know if these plants necessarily have symptoms. Record the GPS coordinate of such a plant. Here the researcher is still collecting the material, taking mainly phloem-rich material and then placing that into a labeled plastic bag. Now to collect the insects, this is using the DVAC. It's basically a leaf blower which has been inverted so that it sucks. We have a stocking at the end that's taken off with all the contents that have been sucked up. Photograph is taken of the number of the accession. This would be directly after collecting the, this will be followed directly by the collecting of the plant material. And then the contents of the sock is poured into the alcohol to preserve any potential vector that might be there. These samples would then be taken to the laboratory and tested for the Aster Yellows phytoplasma.